Okay, everybody, thank you all for joining us today for the first seminar of our fall seminar series. It's great to have so many of you here, so thank you. I do have a few introductions and announcements that we wanted to start with. Since it's the beginning of the semester, I thought we might have several of our new people here. Do we have any brand new integrated plant and soil science grad students present? Oh, I see one small hand. Brian, please stand up and say who you are and who you're working with. Yep, welcome, Brian. And we have at least one new staff member here. Derek, if you want to stand up and say who you, yes, that's a rhetorical question. Please stand up. Yep, welcome, Derek. Oh, another one. That's right, welcome, Josh. Any other new people or grad students? Okay, well, we'll keep that up for a little bit. All right, the other small announcement that I had is that I was informed one of my emails went into a junk folder this week, so I just wanted to make sure that I took this chance to tell you all. We are opting to delete our old graduate degree programs. So you guys all approved basically removing them from the record a while back, but it has since come to our attention that it might be more expedient for just to remove and delete them at the same time. So I sent an email to you all. If anyone opposes that deletion, I need to hear from you by Monday. Otherwise, we'll be unanimous and it'll move forward. We will no longer have those degree programs on the books. All right, and then the very last one is next week is Jason Unrein, so please come back for the second of our 4 promotion seminars that we have going on right now. Today, it is my pleasure to introduce Dr. Erin Haramoto to you. I'm gonna give you a very short intro. Erin came to us straight out of her PhD in horticultural science from Michigan State University in 2014, and so she's been with us for a little over 5 years, 6 years now, along those lines. She was hired as a 65% research and 35% instruction distribution of effort, and her 5-year mean has been 68% research, 30% teaching, and 2% service. And today she's going to give us an overview of what all she's achieved in her 5 years since she's been here. We'll have time for questions when she finishes, and then I will respectfully ask that everybody except the plant and soil science faculty leave the room, Erin included, and we'll have a discussion about her portfolio at that point in time. It's all yours.
I will definitely be leaving the room at that point. Okay, well, thank you, Rebecca, for the introduction, and thank you all for being here today on our first seminar of the fall semester. A brief outline of the seminar today. I'll give a very short personal introduction and try not to replicate what Rebecca said about my background. We're gonna just take a quick look at integrated weed management, why it's so important these days, and then I'll talk about my research program. So first, who am I and how did I get here? Not a rhetorical question. So I did start just about 5 years ago, did receive my PhD from Michigan State, where I worked with a lot of the same management practices that I work with now. So I worked with strip tillage there, now I work with no-till. I worked with cover crops there, I still work with cover crops here. These were in primarily sweet corn and cabbage production systems. For those of you that don't know, Michigan is a really big vegetable and fruit producing state. So I measured, again, a lot of the same things that I look at here in my research. Weed density, weed management. I also had some nutrient loss and profitability components to my PhD. So for context, for my publications list, I came here right after my PhD, but I did a lot of things between my undergrad and my PhD program. So prior to starting at Michigan State, I was a laboratory instructor at a small private college in New England. Prior to that, I worked as a technician for an ARS unit in Urbana-Champaign. And that's where I cut my teeth on corn and soybeans. Lots of corn and soybeans in East Central Illinois. Prior to that, essentially, I received my master's degree. This was now 15 years ago. From the University of Maine, working primarily again in vegetable systems and also with cover crops. And then you will see a couple of publications on my CV that don't look like the rest of my publications. So I was a research technician at one of the University of Maryland labs on the Chesapeake Bay for a few years, right out of undergrad, where I looked at phytoplankton blooms. So my DOE has changed. I was hired on a 65-35 research teaching DOE. We know that these are a little different in your first year. So this information also is in my CV. My first year, I taught one course, integrated weed management, and then in 16 and 17 added one more course per year. So it's more reflective of my actual DOE now. And we, just in the last 2 years, have had these 5% service appointments added to our DOE. So again, that has changed. That 18 year was the first year that we had those in our DOEs. So this will kind of even out over the next few years in terms of research, teaching, and service. I did want to just start too by touching on some of the total funds that I've received since my appointment here. Mostly, or actually these are all competitive funds. And so I've been successful in pursuing funding from a wide range of sources. So from federal grants funded by primarily the USDA, state competitive grants funded by a lot of the commodity boards, and a little bit of industry funding as well. You'll see in my CV, I have a lot of collaborative projects. So just, all right, I'm gonna try to use this here. So the sum total of funding, I can't make a smaller circle, is really high. And that's because we were awarded a $10 million grant earlier this year through the Sustainable Agricultural Systems arm of AFRI. It's a coordinated agricultural projects grant. And this is with numerous PIs across numerous institutions, which I am just one. Dr. Poffenbarger is also on that project. And so this is a huge project. We in Kentucky will be doing 2 common experiments that are going to be replicated throughout the United States, looking at cover crop impacts on soil nitrogen dynamics and pest management. So I'm actually the weed team leader for the one that's focused on pest management. And I'm also one of 7 PIs that are involved in the education component of this grant. So we're going to try to use cover crops to facilitate more active learning in our undergraduate curricula and use these as a systems level look, or as an example of a way that we can be promoting systems level education on agroecosystems. So that number is high, but again, one project forms a lot of that number. And then specifically to my program, just over $900,000. And if you break this down, I know it's a lot of numbers, but I wanted to make one point with this figure. If you break down that $900,000 or so, about a third of that comes from projects on which I am the lead PI, and then 2/3 come from projects on which I am the co-PI. Again, all this information is in my CV. So publications, the figure here on the left is just an updated version of what's in my CV. That was from July, I just updated it earlier this week, looking at the number of times my publications have been cited over the last, well, almost 2 decades now. So in terms of relevant publications, those dealing with agriculture, so not those including phytoplankton, I have 18 that are either published or in press, and 1 that's currently accepted and is being revised. So from Kentucky work, I have 5 that are either published or in press right now, 1 that, again, that's accepted and being revised, and 4 that are still in the submission process, have been submitted and are being reviewed. So this is something I'm really proud of. Of those 10 that are either accepted or in review right now, 6 of those are from graduate students. 5 of those are graduate student lead author papers. And then I'm the first author on 4 of those. 4 of those 10. And so that number there, the 5, 1, and 4, does not include some work that I published since I started at UK, but that was from research that I completed at Michigan State as part of my doctoral work. Okay, so for teaching, again, somewhere around low 30s to mid 30s DOE for teaching. I teach integrated weed management. I've taught that 5 times now. I also teach a graduate section of that course. I have taught our agronomy capstone course 3 times. We didn't have enough students for it to make in spring of 19, but as we get our new degree program online, hoping to be able to maintain teaching in this capstone course. I also teach PLS 531, which is our field school and crop pest management. I teach that in the fall. I've taught that 3 times now, and this has since moved to an alternate year course, so we can try to get enough students to make this, have this make every other year. So this will be fall of even years now. And then I will start teaching the agroecology course. This is a sustainable agriculture. Well, it's in the sustainable agriculture and community food systems major now in spring of 2020, and I'll be doing that in alternate years. So this will be a new course in this academic year. You can read a lot about my teaching philosophy and my teaching statement, kind of why I structure my courses the way I do, and what I try to get my students to, how I try to get them to think and perform in these classes. So I really try to promote critical thinking, connections with material that they're getting, other material from my course, and other courses that they're getting in their education here. So we talk a lot about how plant population density affects weed crop competition, how soil nitrogen dynamics affect weed crop competition, things like that. I try to get them to be self-directed, try and have open-ended learning options for them, and really focus on understanding and not fact recall. And I evaluate these latter two primarily through my use of open book, take home, very open-ended essay questions for exams. So in terms of advising, I have 2.5 current graduate students. Sorry, Cynthia, you're not actually half of a person, but so Austin Sherman has 1 paper in press and 1 paper submitted. He defended in May and started a job immediately as an agronomic consultant. So he's currently completing his thesis. Ryan Collins started in the spring of I can't even remember, 18 was last year, yeah, and is expected to graduate in May of 2020. And so we're gonna hear a lot about the research that both of these students either have done or are doing right now. And then I co-advise Cynthia, she is the half, with Monse, and we're gonna hear about some of her research as well. She will be defending and graduating this December. And she also has 1 paper submitted, and with our collaborators on the project she's working on, 3 total in preparation. And then Tori Stanton graduated from my program and is still working with our group, and she has 1 paper published in weed technology and 2 more that are in the submission phase. So I either serve on or have served on 7 master's and PhD level graduate committees, mostly in IPSS, but 1 entomology student. I advised a Shelgren Fellow in the spring of 2018. That's Michaeli Kofer. She's an ABMT undergraduate. She liked it so much that she signed on to do the UK REC Research and Extension Experience Internship Program this past year. Last year, I advised an undergraduate from Western Kentucky, Gustavo Silva. And then I've also had 3 visiting interns from Argentina and Brazil. So we have Renan with us here this year. And we had Paula last year, she's from Brazil. And then Vinicius was a PhD student in Brazil who's since finished. He was with us in 2017. Okay, so that kind of wraps up some of the context for personal stuff and things that you'll see on my CV. And I'm gonna segue now into talking about weeds, which is why I'm here. So what is integrated weed management? Why do we need integrated weed management? This slide illustrates why we need integrated weed management now. So this is a soybean field. You can see the soybeans in here from Trigg County, Kentucky, soybeans that we took in the fall of 2015. And you also see a lot of weeds, mature weeds that are setting seed. The grower in this field did apply their kind of normal herbicide program in this year, and it failed. And it failed because these weeds have now evolved resistance to that grower's standard herbicide program. So not only do we have this that growers are dealing with kind of on a year-to-year basis, but over time, we've documented an increase over time in the number of species that are resistant to multiple sites of action of herbicides. And so these sites of action are how that chemical works to kill that plant. And so we have upwards of 55 species that are resistant to 2 different sites of action. And we even have some 5 or 6 species that are resistant to 5 different sites of action. So this could, if it keeps going, result in all of the sites of action that we can use with any given crop without causing crop damage or too much carryover issues, having weeds that are resistant to all of them. Another important thing to note in this equation is that we haven't had any new herbicide sites of action released since the 1980s. So discovery is not keeping up with the needs for chemical management. And so the Weed Science Society of America and other professional societies have really put out this call that we need more integrated tactics to help deal with these weeds if chemical management is going to become more difficult or even impossible, which hasn't happened yet, but might happen in the future. So integrated weed management is just using alternative tactics, whether they might be physical tactics, biological tactics, cultural tactics, to help achieve the following goals. And so these are really reducing the density of weeds that are in a field. So we have fewer weeds to kill with the herbicides that we can still use, and the less chance that the resistant allele is going to be out there in a population. So just population genetics tells us if we have a 1 in 10 million chance of having that resistant allele, we have 10 million weeds in the field, it might be out there. If we only have 1 million weeds in the field, we have a lower chance. And then also, so reducing weed density and reducing weed growth too. So keeping weeds smaller and easier to kill using other tactics, including herbicides, again, that are still effective. And then also reducing seed production as well. So reducing the number of propagules that can further these populations. And so most of my research you'll see today and from my CV looks at the alternative tactic of using cover crops, which can provide some different, as we'll see today, a lot of different ways of managing weeds. And the goal of these integrated weed management tactics that go into these integrated weed management programs is usually not to replace herbicides. Herbicides are going to remain likely the primary tactic that we use to control weeds in most of our agronomic cropping systems. But using these integrated practices like cover cropping, we can use them to improve the efficiency and the efficacy and kind of lengthen the lifetime of the products that we still have, the chemical products that we still have to control these weeds. So those of you that work in the more agronomic sections of our department likely know what cover crops are. They're basically things that we plant kind of outside of our cash crop windows. So if we're talking about corn and soybean, we harvest corn in, well, depends on the year, but sometime in September, early October. And then we can plant these cover crops after we harvest corn. We let them grow over the winter. They provide a number of services. They can provide disadvantages too. But then we kill them the following spring when we get ready to plant our next crop. And so traditionally, we've left all that biomass in the field, trying to reap the benefits of having those plants there. But we're kind of looking at some alternative uses for cover crops now as well. But you can see from these photos on the top here, these were taken at Spindletop in early January. This picture on the left is a cover crop, lots of green material there. The picture on the right here does not have a cover crop. So all you see a few weeds here, and then the previous year's crop residue. So one of the things that cover crops are really good for is holding the soil in place, helping reduce erosion potential. This is especially important in a state like Kentucky where we have a lot of rolling hills. And then they also help with weed management and provide other benefits as well. And then these same plots in mid-April, these little plants grow to be these massively big plants. The few weeds that you see here really take over this plot. So when we get to this time, we're going to be terminating this cover crop and planting into that material. So they're just part of our agroecosystems, right? So they have all these other benefits and some disadvantages as well. And so one of the unique aspects of my research program is that with the help of a lot of collaborators, we look at kind of this whole, I'm going to say whole system focus, it's really not, but we look at a number of different aspects of these agroecosystems and how these cover crops influence a lot of those. Keeping in mind that what might be optimal for weed control might not be optimal for other parts of this cropping system. So I'm going to go into instructor mode just for a little bit here. We're going to kind of step through how we can use these cover crops to manage weeds. In our corn and soybean rotations, we primarily use small grain cover crops like cereal rye, like wheat, like triticale maybe. These are more flexible in terms of their planting time. The seed is relatively inexpensive. They're easy to plant, and they can produce a lot of biomass in the fall and spring if you want them to. So most of my talk, I think all of the talk today is going to focus on small grains, and I'm going to be talking about cereal rye a lot. So again, we plant these in the fall, they grow over the winter into the early spring, and then we kill them, usually using an herbicide application. Then we plant our cash crop, whether that's corn, soybeans, tobacco. Again, I'm focusing on agronomic crops, but cover crops are also common in vegetable production systems as well as in grazing systems too. So since we're mostly a no-till state, after we kill that cover crop, all of that plant material forms a residue mulch layer on the soil surface. And both while the cover crop is growing and after it's been killed and as that residue layer, there are impacts on the soil environment. So how much light reaches the soil surface, moisture dynamics, nitrogen dynamics, temperature fluctuations, lots of changes to the soil environment while these plants are growing and after they're dead. And a lot of these factors in the soil then, changes to the soil environment, impact a lot of our parts of our weed life cycle. So seed dormancy and seed germination and emergence and plant growth and reproduction. So while the cover crop is growing, it's mostly targeting winter weeds, those that emerge, germinate and or emerge in the fall. After it's terminated, while it's this residue layer on the soil surface, we're talking about weeds that emerge in the spring and summer, later in the spring and summer. And these are the ones that are going to be competing with most of our agronomic cash crops. So the goals then, if we think about what we're trying to manage by changing the soil environment, it's suppressing seed germination, weed seed germination, reducing seedling emergence, and both of these result in having fewer weeds. So goal number one of integrated weed management, reduce density. And then maybe by altering resource availability, by making a better habitat for some of these seedling or plant diseases, we might be reducing plant growth, which results in reduced weed growth, which was the second goal of our integrated weed management. So a lot more mechanisms exist. There's a lot of other changes to the soil environment, changes to different aspects of the weed's life cycle, but this is just mostly what I'm going to focus on today, a relatively simplified look. So these are all kind of general mechanisms, but if we want to use these to target really specific weeds, specific problematic species, then we need to know something about that species biology. So for example, if we want to be suppressing seed germination by using cover crops, we need to know, is that seed dormant or not? What cues will break the dormancy? What cues promote germination of that species' seeds? If we want to reduce seedling emergence, we need to know, first of all, what phase are we talking about here? When does that weed emerge? Does it emerge in the fall? Is it a winter annual or is it a summer annual? That can lead to changes in how we might manage the cover crop to maximize our benefits. And then if we're looking at reducing plant growth and reproduction, is that weed species even responsive to nitrogen or not? If we're going to be thinking about changes to the soil environment, how competitive is it for moisture? How competitive is it for light? How much does it need light to grow? These are all aspects then about weed biology that can inform how we might manage cover crops to better target key problematic species. Okay, so brief introduction as to why this type of research is important, just kind of a little bit on the dynamics that we study in some of our projects. So now I'd like to give just a few example slides of kind of the breadth of my program. Again, I've done a lot of projects since I got here, a lot of really collaborative projects. So I'd like to just touch on some of those as examples of the kinds of collaborative research that we do. And then later we'll go into more depth on this kind of interaction between weed biology and the environment, and use that as an example of better informing management practices. So one of the first trials I started doing here was with Bob Pierce, looking at cover crops and soil residual herbicides for no-till tobacco production. So we know that increasing the amount of cover crop residue that's left after that cover crop is killed can lead to better weed suppression. You just have a thicker mulch layer, so if you have a light-sensitive seed, less light is reaching the soil surface, less chances it's going to germinate. More physical impedance for that seedling to get up through that thicker mulch layer. But the more mulch that we have, and here's our mulch layer down here, maybe we might be intercepting soil residual herbicides. So these are herbicides that we spray over the top, but they need to reach the soil where they work to prevent weeds from emerging. And these really form the background, or the basis, I guess, for weed management in tobacco. There are very few products that you can apply to tobacco after the tobacco is already up and growing. So we want more residue to suppress more weeds, but maybe we don't want more residue if it's going to interfere with our herbicide activity. So this experiment asked if cover crop residue management affected weed suppression, and so we set up different treatments, either removing all of the cover crop biomass, like we were going to take it out, like we were treating it like a forage crop. We terminated it, or we terminated it early, so there was just a smaller residue layer, or we terminated it later, so there was a thicker residue layer. And we also had treatments with and without soil residual herbicides to see if they remained effective with that range of residue that was on the soil. So we did this on tobacco production farms. I'm going to show data from 3 of those today. These are on the horizontal side here. So farm 1, 2, and 3. And then we measured, one of the things we measured was mid-season weed density in tobacco. So in tobacco, when your soil residual herbicides stop being effective, one of the major ways to remove weeds is either through cultivation, which is not compatible with no-till production, or with a hoe. So helping to reduce this mid-season density is a benefit. So as we're moving, we have our 3 different residue treatments in the 3 bars on each farm. And as we move from the blue, which is removing all the residue for forage, the green is the early terminated, so not much residue. And then the purple is a lot of residue, so a late killed cover crop. We did in fact reduce weed density at 3 of our farms. So the answer to the first question is yes, the more cover crop biomass you have on the soil surface, the fewer weeds you have. We also didn't find any evidence that the amount of residue was affecting the efficacy of these soil residual herbicides. Now, I do want to point out that Bob is very good at timing these applications, and so they always got on not long before a good rainfall event. And those rainfall events can move product that might be sitting on top of the residue down into the soil layer. So one caveat about that, we didn't really test this in really dry conditions. Another caveat is that the 4th site year that I'm not showing here, we had a crop failure. So if you think again about the benefits and potential disadvantages of using these cover crops, not being able to plant into a lot of residue is a pretty serious disadvantage. So a good example of an applied project, one in which we saw some benefits, but we also did demonstrate a disadvantage as well. So one of the trials I have been working on with Hannah is looking at how cover crops impact soybean nitrogen fixation. So again, if we're thinking about these small grain cover crops, they have relatively high carbon to nitrogen ratios. They take up a lot of soil nitrogen while they're growing, and they also immobilize soil nitrogen as that residue is degrading. And so we're talking about both phases of our cover crop here, while it's growing and as a residue on the soil surface. We also know that soybeans grown in soil without much nitrogen in it will fix more of their own. So they will, through biological nitrogen fixation happening in the nodules in their roots, rather than taking up the limited nitrogen that's in the soil. So this experiment asked if soybeans planted after a cereal rye cover crop do indeed derive more of their plant nitrogen from this biological fixation relative to those relative to soybeans planted into fallow or bare soil. So the first thing Hannah looked at was the soil nitrogen. And so indeed, following that cereal rye cover crop, we did draw down the soil nitrogen compared to soil nitrogen following no cover crop. And then she looked at the percentage of total nitrogen contained in the soybeans that was derived from biological fixation. And yeah, it was higher following the cover crop than following the fallow treatment. So the answer to the question was yes, they do. And we have another experiment currently in the ground, and we'll be planting cover crops for next year's soybeans too, looking at this in greater detail. And then this has led also to a project with Montse and others in this department and other institutions looking at how this might influence protein accumulation in soybean seeds. One more example to talk about kind of the breadth of this program, the program that I have here looking at cover crops. With Montse and with Cynthia, the graduate student that we co-advise, we're part of a large network. Well, we have a number of PIs looking at this in Nebraska and Ohio and here in Kentucky. And we're asking the following questions. So if we plant cover crops earlier in the fall, we can increase biomass production. But if we still have, say, corn or soybeans in the field and we can't harvest them yet, it's much more difficult to plant that cover crop. So this experiment essentially asks if we can plant shorter season soybeans, so a lower maturity group, to facilitate, that are harvested earlier in the season to facilitate earlier planting of cover crops. And then if we can, with various cover crop planting dates, how much biomass do we generate, and what are the impacts on weed suppression. So this is just a couple aspects of this large trial. I've borrowed this great diagram from Cynthia that she uses to explain this, the outline of this experiment. So all of our soybeans are planted on the same day in the spring. But we plant long season soybeans. So for those of you familiar with this terminology, this is like a maturity group 4 for us. Maturity groups define kind of the length, well, they define a lot of things, but one of the things that they determine is the length of the soybean growing season. So these are harvested though in our area in mid-October. So we can't plant our cover crop to until the middle of October. And that limits the potential biomass accumulation. So if we're planting shorter season soybeans, again, they get planted at the same time, but they take less time to mature. So we're harvesting them earlier. And for us, a maturity group 1 soybean, we harvest in around this time of the year. So we can plant our cover crops earlier in the fall. So one advantage of this is that we can get more cover crop biomass and better weed suppression, particularly of problematic species like we'll hear about later today, like mare's tail. But one potential limitation is that these shorter season soybeans are likely going to yield less. So we really have to find a balance then between what maturity group will lead us to having comparable yields versus what is going to provide us with enough time for adequate cover crop, ground cover, and biomass production. So again, this is a massively huge trial. We have a lot of data collection going on, and Cynthia's done a really good job of distilling this down and looking at this from a number of different perspectives. So the first figure that I'm going to show is looking at these maturity group soybeans. So the higher the number, the longer the growing season, the shorter the number, the smaller the number, the shorter the growing season. And so she's compared the yields, the soybean yields across those different maturity groups. And you do see that as you have shorter season soybeans, they yield less than the longer season soybeans. But, and this is, she's also done, submitted a paper to Field Crops Research with a lot of modeling across our different sites, different weather conditions, and using 30-year climate data as well to look at the potential for these different maturity groups to yield under those conditions. So if you're really interested in cover crops, and we'll see why, this difference between these maturity group 3 soybeans and 4 is not really all that great. It ends up being about 300 kilograms per hectare difference in yield, which sounds like a lot, but just hold on. What she also found though, is that planting, looking at the cover crop biomass production, following these different lengths of the soybean growing season, if you do want to plant a 3 versus a 4, you get a lot more cover crop biomass in the fall. So again, each individual grower needs to decide, is that worth it for me or not? And so this is not just cover crop biomass, but she also looked at the percent ground cover produced by those cover crops. And the maturity group 3 is in these blue symbols, the maturity group 4 is in these black symbols. And if you look at ground cover over our late fall / early winter period, there's a huge difference in how much ground we're covering with that 2-week difference in cover crop planting date. So this is a good, ground cover leads to better protection from erosion. Ground cover can also lead to suppressing weeds. So depending on your goals here, depending on what you want to get out of your whole system, this is a good example of kind of the trade-offs inherent in cover cropping versus maximizing yields. And this, I'm just showing data from 1 year of this trial. We're just now wrapping up this trial and have a couple other papers that are being prepared on more of the cover crop and ground cover data and weed suppression. Okay, so that gives just an idea of the different types of projects that I do, collaborative projects. And now I'm going to go into a little bit deeper dive into learning both the research that we do to learn more about mare's tail so that we can kill it better. So kind of taking this approach of biology, how the weeds interact with the environment, better informing our management practices. And here's mare's tail. So this is a weed in the Asteraceae family, Erigeron or Coniza canadensis. It is actually a native species to North America, unlike some of our agronomic weeds. And it used to mostly be found in disturbed areas. but it's now a major pest of no-till soybeans in particular. So Carol Baskin and Jerry Baskin have done a lot of work classifying seeds and seed germination, dormancy and germination ecology. And so from their book on seeds and some of the other work that they've done, the achenes, they're not quite seeds, are shown in this photo here, and they're generally considered to not be dormant. So what that means is they will germinate if environmental conditions are right. There is some populations might have a small fraction of seeds produced that have non-deep physiological dormancy. So they won't germinate if conditions are right, but it's a really broad set of cues that will break that seed dormancy. So mare's tail is classified as a winter annual, which normally would mean it emerges in the fall, grows through the fall, winter, and spring, and flowers in the spring. But spring germination can also occur, especially, we think, the further south you go. And this is going to be a major theme of the next few slides. And so in terms of the cues that this species needs for germination, it does require light. It does require certain wavelengths of light. It requires soil moisture, and the base temperature for germination is around 12 degrees Celsius. So why is this weed such a problem? Because in part of that small seed, I forgot to point out the scale bars are about those seeds are about 1 millimeter long without the pappus. So they can have really high emergence percentages, germination and emergence percentages in no-till systems. As soon as you bury that seed, it doesn't get that light cue it needs for germination, and it won't germinate and emerge. This variable emergence timing is also really problematic. Is this weed going to emerge in the fall, or is it going to emerge in the spring? We don't really have a good way to predict that yet. It's very competitive. So if it's completely uncontrolled, we can have upwards of 90% yield loss. This photo shows a soybean field. I took this last weekend outside of Millersburg. This is not an example of a complete control failure. So this field is not likely to have 70 to 90% yield loss. But mare's tail also has herbicide resistance. So glyphosate resistance is pretty common, or can be common in Kentucky. It also has some tolerance. So that means if you spray this weed when it's too big, for example, or when conditions are not quite right, it might survive that application and be harder, either require a repeat application or just be harder to manage in general. So the life cycle of this weed, it's out there reproducing and shedding its achenes right now, end of August, early September for us in Kentucky. And then those seeds, again, small seeds, form these really small seedlings, about 2 millimeters across. People that work with me and myself, we're getting very good at distinguishing all of these tiny little seedlings in the field. But the timing of this, again, the conditions need to be right for it to germinate. Is this going to happen in the fall or the spring? Once those seedlings that do survive grow to be small rosettes, fall emerging plants, this is their overwintering stage. And then as the season continues, they just keep getting bigger and bigger, they bolt, and then, reproduce, and the cycle continues. So one of the questions that we have with our research is, again, this timing issue of germination and emergence. Is this going to happen in the fall, or is this going to happen in the spring? And we address this with the next couple of studies are part of an AFRI-funded trial, a collaborative trial that I have with folks at Southern Illinois, Kansas State, and Missouri. This is the trial that Ryan's working on for his master's degree. And we're asking, so what factors regulate the emergence time? Soil conditions, environmental conditions. And then if we can nail those down, can we better predict when mare's tail is going to emerge? So the emergence timing has a really big impact on how you manage this weed. So these are two photos taken on the same day at Woodford Farm in May of 2017. So the plant on the left is 6 inches tall, 6 to 8 inches in diameter. So that likely emerged in the fall. But you see these two tiny ones here, these same day, same pen for scale, emerged in the spring. So two questions you can ask with this. Which plant would you rather kill prior to planting soybeans? Probably these. Your chemical applications are going to be more effective. And then for plants that are this big, would you rather spray them, treat them in the fall when they're this size and you have a better likelihood of controlling them, Or would you rather wait until it's time to plant soybeans and try to kill them when they're this big? So emergence time has a large impact on its management. So we have a common garden experiment looking at this emergence time question. The first year that we did this, we plant mare's tail from 8 different populations across our 4 states in each state. We track emergence bi-weekly. The first year we did it, most of our mare's tail emerged in the spring. Here's April, March and April. But this was 16-17. We had an incredibly dry fall in the fall of 2016, very dry. So we can kind of guess from our weather data, our precipitation and temperature data, air temperature data, that maybe this is going to be important. We are also collecting soil temperature and soil moisture data as well in support of, again, trying to build these models. So first year, spring emergence. Second year we did this trial, almost all of it emerged in the fall, almost as soon as we planted it. So this fall was very wet. Fall of 17, this was one of our tropical systems that came through, and we had almost 30 centimeters of precipitation between when we planted the seed and when the soil temperatures dropped below the base temperature for germination. So it's likely that soil moisture and temperature, again, we have these data, we're working on developing these models, are going to be important in predicting when mare's tail is going to emerge. But it is important to note too that this combination of factors doesn't predict emergence of a lot of our other winter annual weeds. So mare's tail would be relatively unique in this aspect. And so again, these data, both the emergence data, our knowledge about the biology, the germination, dormancy and germination ecology of this, and then the soil temperature, moisture, precipitation, air temperature data, we're hoping that all of these will help us build a better predictive models across all of the study locations that we're looking at. So next question, for these fall emerged plants that are overwintering as this size here, what factors control their overwintering success? So we've noticed that in some years we can have a lot of mortality of these fall emerged plants. In some years we can have almost no mortality of these overwintering plants, or these fall emerged plants. So the second question then is, how much does overwinter mortality help to reduce density if the plants are going to emerge in the fall? And so to answer this question, we measured the diameter of 100 rosettes in each state in the fall. We mark them so we can come back to each individual plant in the spring. And we measure whether or not we look at whether or not it survived and measure the size. So eventually these will all be used in a survivor analysis and a time to transition analysis, again, across all 8 of our site years. So I'm just going to show data from this past winter from Illinois and Kentucky. I created a histogram here of the initial rosette diameter, and then the frequency of plants that fell into each one of those size classes. And it does have some skews. It's also slightly bimodal, but anyways, initial rosette diameter. And you see that same histogram in the background here with this darker green, but now I've overlaid in the lighter green a histogram, again, based on the initial rosette diameter of whether those plants survived. So for our larger plants, we had a higher percentage survive this past winter. For our smaller plants, a lower percentage survived this past winter. This is the second winter that we've done this trial. The first winter, all of our plants died. What happened to them? You see this rosette right here. You can also see how wet the soil is. And then this right here is about 1 inch of exposed root. So they frost heaved, essentially. And this, again, is 17-18, all of our plants did this. So what are we going to do with these data? Well, one, we'll have a better understanding, again, with our soil moisture, soil temperature data, precipitation, snow cover, air temperature data as well, of understanding winter as a source of mortality. So can we model the survival based on all of these factors and initial plant size as well? And it also leads to questions about how important frost heaving is as a source of mortality based on these soil conditions, and whether plants are protected from this frost heaving if they're growing underneath the cover crop or not. So you'll kind of notice a theme here of biology of that plant, so the dormancy and germination information that we know about those, weather and soil conditions. So emergence times appear to be affected by, so some of those dormancy and germination cues, availability of those, specific weather conditions in the year, and specific soil conditions in the year. which are impacted by weather. The overwintering success seems to be affected by initial plant size, and again, specific weather conditions, and their impact on soil conditions. So what's unique about this project is that we're measuring kind of the interaction between all of these different factors, again, with the goal to try to develop these predictive models that we can use to inform our management decisions. So let's talk about management and killing mare's tail. So regardless of whether that plant emerges in the fall or in the spring, how do cover crops contribute to managing this weed both prior to and in soybean? And so we're going to transition now from Ryan's project into a project that Austin Sherman did with JD and I at the Woodford farm. His project involved a factorial combination of a cereal rye cover crop, so with and without cover crop, with and without a fall herbicide application to target those weeds that might be emerging in the fall, and with and without an early spring herbicide application. That was applied before the cover crop was terminated. So we're going to just focus in on 2 of these factors today. And so as part of his project, Austin went out frequently and counted mare's tail density. And again, this is something that a lot of projects don't report on. It's the really fine scale look of changes in density through time as affected by these different management practices. So the first year of this trial, 16-17, at this location, we had a lot of fall emerged mare's tail. And a really good cover crop stand over this fall and into the following spring. So just taking a look at data, I'm only going to present data from before soybean planting, about a month and a half before soybean planting, so when growers might be having to make decisions on what herbicides to apply. And then within a week or 10 days of soybean planting in each year. So we're going to look at mare's tail density at both of those times. So in the first year he did his trial, as you might expect, and then we're going to look at 4 treatments. So with cover crop are shown in green symbols, without the cover crop are shown in purple. Ones with that spring herbicide application are triangles, and then the circles are without that spring herbicide application. So as you might expect, when you do nothing, you have no cover crop, no herbicide application, you have a lot more plants in the field. Where we had either that spring herbicide application or a cover crop in the field, density was a lot lower, as we might expect. So as we get on in time through our soybean planting time, first thing to point out is that the treatment that only had that early spring herbicide application, density of mare's tail increased by quite a bit here. So that herbicide will kill the plants that are out there, but it does not prevent additional emergence. So this is our spring flush of emergence here. Most of our plants emerged in the fall, but we still had some come in the spring. But any treatments we had that had the cover crop, regardless of whether or not they had that spring herbicide application, had really low mare's tail density. So in this year, where we had a good cover crop stand, that plus its residue did prevent that additional spring flush of emergence. As you might guess, I'm going to show you the second year of data now. And as is what makes field research so fun is that you never get 2 years that are similar. So in the second year, remember the first year, mostly fall emergence, second year, spring emergence, and late spring emergence too. Winter of 17-18 was a hard winter. We had a lot of moisture, and we had really cool temperatures through March and April as well. So we think this led to the fact that he had a poor cover crop stand in this year. So just about half as much biomass produced in this year relative to the previous year. It's going to be different. Before soybean planting, though, the densities were relatively similar. So higher densities where we had no treatment, and then where we had either the cover crop or the spring herbicide, lower densities. It was really after planting, though, that differences became apparent. So again, we had a large flush of emergence. So where we had the herbicide only, there was nothing there to keep the mare's tail from emerging. The other major difference in this year was that the cover crop also was not effective in suppressing that emergence. So we had a poor stand, less biomass, and then a lot of plants emerging at this time. And so that combination of factors, we think, led to the cover crop not effectively controlling this emergent flush in this year. So 2 years of the data side by side, a couple of key similarities that we can draw from all of this is that in both years, the cover crop did reduce mare's tail density. It just happened to be more effective, or mare's tail density right at soybean planting, within a week or so of soybean planting. It just happened to be more effective in the year where we had more biomass. It was not as effective in the year where we didn't have as much biomass. Now, we can't really tease apart the role of the biomass versus that spring emergence flush quite yet. I have to do another study to figure that out. So the other similarity between our 2 years, this early herbicide application kills weeds that are out there. Because of the product that we used, it doesn't prevent additional emergence. Now, we could apply a soil residual herbicide at this time, but we want to maybe save those products for weeds that are going to be emerging later on in the season too. So if you have Palmer amaranth or water hemp, for example, you might want to save those products to use to control those weeds. If we can manage mare's tail with the cover crop, that might be beneficial for us. I'm just going to skip ahead here, and I'm going to skip these in the interest of time. Okay, so tying together all of this mare's tail research. So again, a lot of the research looking at emergence time and overwintering success really highlights the importance of knowing how that species, how its seeds are going to respond, how its seeds are going to act, how they're going to respond to differences in weather and differences in the soil condition. And those two are really tightly linked together. So we did show that cover crops can suppress mare's tail, and their success in suppressing mare's tail until we planted our soybeans depends on the emergence time of the mare's tail and the cover crop biomass. These are the two factors that really differed in the 2 years of our study, of this study. But keep in mind too that cover crops are living plants. So they're also impacted by the weather and the soil conditions in any given year. And also, as just part of this total system, optimizing cover crops for mare's tail control might result in disadvantages showing up in other parts of our production systems. So really, our research is trying to help inform management decisions. Based on the density of weeds and based on the success of the cover crop stand, we're hoping to be able to provide better recommendations for when growers need to be thinking about managing mare's tail. And by population density, this should say emergence time and population density. Okay, so I have reached the end of the research section of this talk. We all know that this job is tying together all of these projects and all of our other commitments it can be really stressful, and none of us do it without support. And so I would really like to acknowledge the faculty in the weed science program, specifically acknowledge Matthew Allen, Sarah Carter, James. I don't have a picture of you, James. And numerous Marcy Rucker and Linda Williams and others in the weed science program, in addition to a number of other folks at Spindletop that have helped this program tremendously. Former and current graduate students and postdocs, undergraduates and interns, and all the weed science students that went to the weeds contest this year, and a number of other students that have worked with our research group over the last 5 years. I'd like to thank all of them for their help. My collaborators, I tried to create a collaborator slide, it got out of control, so you know who you are. And so if you've helped me write a paper or write a grant or listen to me complain about the weather or any of these other things, especially if you've become a friend and you'll take me out for a beer, or you help me out after I have knee surgery, or help out in a million cajillion other ways, double thanks to you. We also rely on our family for their support. My family is very small. It's comprised of the 1, 2, 3, 4, 5 people that you see on this slide. My mom has passed away, and my cat. They don't live anywhere near me, but they still provide more support than they probably know. And I want to end with just a couple other slides. For me, when I need to, going outside really helps. We live in an amazing commonwealth, not a state. So I just wanted to share some of the photos of things that I've done over the last 5 years when people have managed to convince me to get away from the farm or off my computer. So get outside. And then lastly, you all know I'm an avid cyclist. I used to be an avid runner, and some of our Bourbon Chase team is in the room today. So being on my bike too has provided a lot of stress relief. For me, over the last 5 years, I've had the highest of highs and the lowest of lows on my bike. I've seen things I never would have thought I would have seen. I think you get the idea. I've been on roads that would take your breath away. And so to my cycling family as well, Thanks for helping to keep me sane, and sometimes literally pulling me up mammoth hills. So with that, I'll leave you with the sunrise over a horse farm taken early because I was going to ride my bike, and the moonrise over Spindletop taken late because I was out riding my bike. And I will ask you for any questions you might have.
Any questions in the room? I'll ask it. You can allow Princeton.
I will repeat your questions as well.
So most of your cover crop work was with rye, or did you do a lot with legumes also?
So the question is whether most of my cover crop work is with rye or whether I did research on legumes as well. I have a couple trials that include legumes. So one of the aspects of the trial I did with Bob that I showed today was looking at cover crop mixtures. So mixtures of small grains with legumes and sometimes other things versus cover crop small grain monocultures. And we also have a large trial that we're doing with mixtures of wheat or cereal rye, both with crimson clover. So the issue with legumes as cover crops in our corn and soybean systems is that they need to be planted relatively early here in Kentucky, like now for some of them. Crimson clover we can plant a little bit later, but getting them established before the winter is one issue. The second issue is that a lot of their benefits that we derive from them, from their biomass production, we have to kill them late in the spring. So they really need to go into April, maybe even May before we terminate them. Most of their biomass accumulation occurs in that mid to late spring period. And most growers do not want to wait to plant. They don't want to delay planting corn, for example. So those are two kind of strikes against using legumes in these production systems. In vegetable production and tobacco production, where you have more flexible harvest and planting dates, they make more sense. Very long answer to a short question.
Any others out there, or ask Princeton if they have any questions.
Princeton, do you have any questions? Princeton?
I don't.
One more, Bob.
So you mentioned with the mare's tail, the lack of the dormancy. So what accounts for this broad range of germination? Is that depth?
So yeah, the question is, if mare's tail doesn't have seed dormancy, how can it emerge in either the fall or the spring? Am I getting to that? Yeah, so seed dormancy, a seed can be non-dormant and still not germinate if it doesn't have the right conditions. That was a lot of knots. So if it's too dry in the fall, for example, the seed doesn't have adequate moisture, it'll wait until the following spring. So that's one issue. And then, I think a lot of it maybe has to do with seed movement around, potentially into new, as we do have these freeze-thaw events occurring, maybe some of those seeds get moved into safe sites that are more, in which they're better able to germinate. So yeah, even though the seed doesn't have dormancy, either primary or secondary dormancy, it still might not germinate if conditions are adequate. Uh-oh, now Carol's gonna chime in. So Carol's asking for spring germinated mare's tail plants, do they flower and produce seed in the same year, or do they overwinter and flower and produce seed the next year? And to my knowledge, they all flower and produce seed that year. So they don't overwinter if they germinate in the spring. Another question.
So I was thinking about this related to the question I was thinking about earlier. So you can germinate in the fall, and it can mature around now, so you call it winter annual, but it's like a full season plant, isn't it?
Yeah, and so it is classified, and it's classified in seeds as a winter annual, but right, the flowering time does make it a little bit more of a summer annual. And so this is still, for them to completely be done reproducing, it still happens earlier than a lot of our true summer annuals, because when we're going out to sample at harvest time, for example, our mare's tail plants are kind of just skeletons. So all of the seeds have been shed and they're completely senesced. So it is kind of a strange, the life cycle of it is strange. And I haven't even looked to see if it's been classified as was summer annual. I'm trying to think what the hybrid of those would be, but it does all reproduce at this time of year. Yeah.
Let me add, here in southern Kentucky and Tennessee, there are a couple of other Asteraceae that do the same kind of thing, Selenium amarum,
Right.
Eudorecia. They both have seeds that are not dormant and they fall off when it's still kind of warm, they'll germinate. Otherwise, they wait and germinate in the spring. So with those, the fall germinating ones we call winter annuals, and the spring germinating ones we call summer annuals.
And from a management perspective, you can argue that it is a summer annual because you need to kill it so that it doesn't flower and produce seed in the fall. And so its classification as a winter annual weed makes sense from some perspectives, but from other perspectives, it kind of doesn't. And then, yeah, do you classify for the same species? Do you classify spring emerging plants as summer annuals, fall emerging plants as winter annuals? It's very confusing. And it's also very confusing for students when you try to teach them that weeds have a life cycle, except sometimes they don't. They break the rules. And yeah, Helianthus annuus no, that's not the one I'm thinking of. Scentless chamomile acts the same up north, where I'm from, can be either. So can Stellaria media. So there are weeds that kind of don't really fall into these easy categories, which makes studying them so much more fun.
What's the seed longevity or viability in the soil seed bank?
Of mare's tail, it's thought to be relatively short. So, sorry, the question was the longevity of seed in the seed bank. It's thought to be relatively short. And we've talked about doing some, so you guys have studied buried seed of it, and it was short. And we've talked about doing some updated studies in a no-till environment on the soil surface too. Next year.
The diameter looked like it played a significant role in survival and temperature, like in the winter. And you said there's different times that are needed.
Yeah.
Could that play a role as if there's different varieties that have different diameters that play into the germination time?
Yeah, so we have not, so the question is about the diameter of overwintering plants and that related to emergence time,
Yeah,
essentially.
There's two varieties, maybe there's a small diameter or germinating.
So the whole biotype question is another interesting part of this equation. So I don't know that there are different biotypes or I'm going to call them biotypes that would germinate in the fall or in the spring. And so previous work in these common garden studies where you take populations from different locations, say on a north-south gradient, and you plant all of those biotypes at each location. So there was a student in Missouri that did this, and he showed that it was in northern Missouri, all the biotypes, regardless of where they came from, still germinated mostly in the fall. And in southern Missouri, regardless of the source population, they mostly germinated in the spring. So if you're trying to tease apart whether that emergence time is related to the genetics of the plant or environmental conditions, those types of experiments suggest it's environmental conditions. If it was biology, if it was the genetics, plants from up north should germinate in the fall, regardless of where you plant them. And that's not what they found. I think, so we don't have, one of the other bits of data that we do need to collect is looking at, the emergence time of these each individuals and then how big they can get. So if they emerge on August 20th and they're like that very first cohort, are they going to be those plants that are 80 millimeters across in late fall when we go to measure them? Most likely, yes. But then maybe something will come along and eat them, because things do like to eat our mare's tail plants in the field.
Okay, guys, we're 5 minutes